Here, I have to say that I want to congratulate the NCBI, especially Jim and David, for having done that most strange and bizarre of things, produced a government business that actually works and does more than was asked of it. Okay, really has been a pleasure to get into this sort of genome business with this kind of backup. And in fact, my talk is not about Genbank. My talk could have been called Beyond Genbank, because as you have ably heard, most recently from Jim, the business about what the genes do was not taken as the remit of Genbank or any of the originating people. For some reason, and something I could never understand, the early advocates of sequencing seemed truly to believe that if you knew the sequence of everything, you would understand how it all worked. Okay? I never believed that. Geneticists were sort of very restricted in their enthusiasm for this point of view. To begin with, this was a view they attributed to biochemists, I should say. But as Rich told you this morning, the real biochemists didn't believe it either, and certainly they don't believe it now. And so the whole question is, how do we put biological understanding into the picture is really the theme of my talk. All of this presupposes that Genbank exists and that it is as excellent as it is. So I want to do a little bit of history of genomics from the point of view of a simple geneticist. Okay? First of all, genomics is not a new word. It's not genomics was introduced, this is from the OED, by Winkler in 1920 or something like that. Okay, Winkler was an embryologist. It still means what it meant then, pretty much what it means now. And there is some difference in this business of prediction. I found this, in the OED found this 1970 Scientific American article about how many genes they expected to find. I always imagined that this must have been a neurobiologist, because they've always had trouble understanding how you can think with a finite number of genes. Now, the history is basically the history of genetics. I've put it into a couple of slides. At first, we were worried about the mechanics of inheritance, and this is now all in the textbooks. Then we were worried about how the DNA information is expressed, and we ended up with the ineffable central dogma. This is basically orthogonally arranged, the same order that Jim had in his schema of Genbank, going from the bottom to the top. Okay? And that's maybe why he, as a biologist, found it natural, because the central dogma was there. In the 1960s, of course, this began to change, and a few pioneers, the phage group, became interested in the question of what do those genes do? And they did this by using conditional lethal mutants, and the theory was that if you got a conditional lethal, it must be essential, and if it's essential, and if the method by which you got conditional lethals was general, you should be able to get such mutants in any gene, and if you saturated the genome with them, you could find out all the functional parts of the genome. Notice that we're going from the top to the bottom here, not from the bottom to the top. Okay? And the phages were attractive, of course, because it was clear already that they had a very small number of genes to worry about. And this is the first genome paper that I could find. You can see it's a genome paper, very many authors, different labs, different continents, and the central author is not the first or last author. It's Edgar. Okay? And on the left, you see a portrait of the organism, and on the right, you see the first genome database. On the outside, on the inside of the ring is the map order done by mapping, and on the outside is a little ideogram. And it is that ideogram that tells you what goes wrong when the gene on the inside is mutant. Okay? That's the essential pieces. And in fact, this was such a powerful idea that a number of other pioneers began to do the same kind of thing with simple eukaryotes that also had smaller numbers of genes, arguably. And they were chosen also because they were easy to manipulate, and you could do experiments in them internally to them without reference to outside experiments. And the founders of these communities had a philosophy of science, best summarized by Delbruck. The idea was that we all work together, we compete with each other, but we share the same information. This was not universally the case in experimental biology. This was something quite remarkable, even for the time. And these, of course, became the model organisms. And when the time came, they were the ones who were sequenced first. So I want then to sort of summarize what the geneticist's view of the essential part of understanding is. Geneticists distinguish between structure, the genotype. That means that basically genetic maps. And the ultimate in a genetic map is the sequence of the DNA, especially if the mutants are on there. And the second thing they're interested in, and actually arguably more interested in, is function. They're pleased to name this phenotype. And that is the biological roles of whatever those elements on the genes that are inherited are. And they can be genes, it's classically defined, sequence motifs, individual bases. And historically, these were inferred from the biological consequences of mutations, white eyes or something. Today, the inference is still, at the end of the day, from knockout mice, okay, and things like that. And these data, unlike the data on the top part of this chart, these data are much more difficult to summarize. Okay, lists of genes or annotations and references to the literature, and these things are stored in databases, by and large not so much at NCBI, although there are some, but in other databases that are designed to understand the information. That is above the sequence, if you like, the phenotype level, as opposed to the genotype level information. I saw this at the Museum of Natural History. They had a Darwin exhibit. He made this sketch in his notebook just after he got off the Beagle. And I think it's one of these great premature insights. I mean, you know, that is pretty close to the way it actually looks. And this idea, of course, has animated biology for a long time, but in molecular biology, it became relevant relatively late. Here are two viruses. This is phage P22. This is phage lambda. They grow on what were regarded as different species of bacteria. They look different, but it turns out they have similar genetic maps. When you look at the maps and you include what the genes do, you discover a striking regularity, namely all the regulatory stuff to a huge level of detail is the same, and then there's DNA and control and lysis and heads and tails and so on, okay? And it turned out that you can make hybrids between these two viruses by genetic manipulations that are only the ancients knew, and when you do that, you get viable things out. So these things have interchangeable parts, different species, allegedly. This is an early genetic map of E. coli, and again, the E. coli people named the genes in such a way that you had some idea of what they did. And I show you this to show you that, in fact, there is a logical progression from map to a higher structure map to the sequence. This is Blattner's annotation of 1 or 2 minutes. And what you see is, yeah, there's a lot of stuff in there, but basically this is just the ultimate in genetic maps from the functional point of view. So the sequences were sequenced, and the sequences went into Genbank, and there was a lot of back and forth about whether the whole sequence could go into Genbank, but only 350 KB, and you heard all about that. I'm going to skip that. And the clear thing, which had been prefigured in earlier literature, is that for these eukaryotes, the basic cellular functions are carried out by proteins whose structure and function are conserved. You can follow them all the way back to the beginning in many cases. That doesn't mean that their regulation is conserved. It doesn't mean a lot of things, but it does mean that a lot of the function is conserved, and I'll show you some example of that. Here's a map of Saccharomyces cerevisiae well before sequencing, and like the coli one, I won't show you a zillion slides, it comes out the same. And of course, humans, it was more complicated, and one had to use restriction polymorphisms and other DNA polymorphisms to figure out mapping, and eventually one got maps that looked like this, but at the end of the day, we got sequences again. Now, what do those sequences mean in the human? Well, in the human, the idea was that there are some diseases like these for which it was obvious from the structure of the inheritance patterns that they are likely to be single Mendelian genes, and there's a list of these here. And so the idea is very simple. If you find the right gene, you will find a correlation between a defect in that gene and the phenotype, and it will be the only gene for which that is true. And that turned out to be the case using these methods. And at the end of the day, you ended up just like in E. coli and just like in phage and just like in yeast, you ended up with a sequence. This happens to be the BRCA2 gene, comes off the Genbank, the NCBI website. And you see there's splicing and there's other various details, but at the end of the day, it's a gene and a protein. And interestingly, most of the, we did this review a while ago, most of the genes that cause Mendelian diseases do change proteins, RNAs and regulation notwithstanding. Only at that time, only 213 were not visibly altering a protein. Now, complex inheritance, on the other hand, is a completely different story. Many of us, me included, very vociferously I should say, thought it was just going to be a couple of genes or 3 genes, that combinatorial complexity and so forth and so on was hiding the fact that this was really a simple proposition. Nothing turns out to be farther from the truth, and I think the fair way to put this is to say that although we keep finding statistically significant, undoubted, reproducible findings, what they amount to in terms of explaining the disease is very small. And we don't understand why. And in fact, one of the great open questions in my mind is having a robust theory of how genes interact to produce phenotypes and a complex inheritance. Here's an example of failure. Tom Marr, who was involved in this along a cast of thousands. And we got lots and lots of exactly the right families to look at bipolar disease, high heritability, simple apparent inheritance, and the conclusion was the results exclude monogenic models and make it unlikely that two genes account for the disease in this sample, 2 independently segregating genes. The answer it really is that we don't have a clue, and it was really hard for the psychiatrists to accept this. It took a very long time to get this published because I wanted to get all the authors in the tent. There's a lot of statistical education involved. Okay. If we're going to talk seriously about sequence and function, we have to confront an actual sequence. Okay? This is a sequence. Right? What can you tell about it? Virtually nothing. Okay? Some of you in the audience either have heard me before or know how I've spent my career in some detail and will guess what this sequence is. But there's no internal information unless you have a terabyte memory that can tell you what this sequence is. Okay? However, if you do a sequence comparison, you compare it you get a human hit, and that human hit, this is the blast hit for this sequence, and it's actin. And the important thing, the burden of my talk here is, how do I know it's actin? By looking at the sequence, I could tell nothing. How do I know it's actin? I know it's actin because at the end of a chain of homology searches, there is a sequence, a protein sequence, on which somebody did an experiment. In this case, it's a rabbit sequence, and somebody figured out that this gene is the gene that hydrolyzes the ATP when your muscles move. It's actin, right? And what you need to remember about all this sequencology, at least in my humble opinion, is at the end of every trail is an experiment, just like Rich Roberts said this morning. Having a sequence gives you a hint, maybe, tells you where to look, but at the end of the day, there has to be an experiment. It doesn't have to be a biochemical experiment, but it has to be an experiment. Okay? In 1988, in response to a particularly ignorant article in the obscure journal Science, Jerry Fink and I were moved to write this article, and this has a sort of Astel-like diagram in it. And basically, it shows you at some epistemological level what the relationship is between genes and proteins and functions, and what that has to, and how these things are inferred with any comfort. And the article here was to say, first of all, that the previous article was mistaken when it thought that yeast was a prokaryote. And it was even more mistaken when it thought that nothing would ever be learned from yeast. This is in the late 1980s, I should say. And the idea of this was that in yeast, you can do the experiments internally in a single system to try to figure out what a particular protein does with maybe the greatest ease for any eukaryote. That was the burden of the argument. Okay? So the kind of thing, this is a table from that paper, the kind of thing that we had in mind was something like this. Now, I've updated the table. You can go back to the original Science. Only about half as many of function things were filled in. But basically, this is the story as it is as of about 10 years ago. There's a bunch of conserved proteins. These proteins are sorted by their degree of amino acid identity. And what this function here is that what function means that when you blow away the yeast gene and you get a phenotype, usually death, and you replace the coding sequence with the mammalian or the human coding sequence, you get the phenotype reversed. That's like saying in the car example, where if you have a Mercedes over here and a Model T over there, Model T doesn't run, you reach into the Mercedes, grab a part, slap it into the Model T, and drive away. Of course, in the 120 or so years of evolution of motor cars, it's hopeless, okay? But this is 2 billion or so years. I don't know what the taxonomists say about the distance between human and yeast. But this works much of the time, especially for these high-ranking hits. Some of these genes are being used in ways that are completely dissimilar. Even actin, after all, is essential in functions that yeast doesn't have, like thinking. I take an inhibitor of this one, okay, and in order to reduce my cholesterol, but the truth of the matter is that yeast don't even make cholesterol. They make some other steroid. But nature doesn't reinvent. And that's really what this is about. Okay? So this was a huge favor to the biology community because we could do an experiment over here in yeast, get an answer that was an even much broader hint about what the protein is doing over here in my cancer cell. Of course, I did do this just to emphasize not to put down the Genbank-like databases, but to show you that they don't mention this here. There are all these tracks, and there's all this homology, but it never tells you what the thing does. For that, you got to go to the literature. And that was the most brilliant thing that NCBI did. They connected, in a really robust way, the sequences with what is known about the sequences through PubMed and all the rest of that sort of stuff. And when we get to PubMed Central, and you can search, on virtually anything, that's going to become even more robust. And that's, in fact, When I say the nature of biological understanding, that's how we work. We get a sequence. The sequence leads us around to papers in lots of different taxa that tell us what RAS does in all of these different circumstances because somebody did an experiment. Here's another. Okay. Now, the next issue that I want to address is that, of course, proteins don't work alone. Protein works in great gangs of assemblies. Actin is, which I happen to work on, a lot of these examples are taken from my own work because I know it very well, and not because I couldn't make examples from 1,000 other people's work that's better than mine, but you know, I'm me. Okay. So actin, which I happen to work on, has very, very many ligands, other proteins that bind to it. And many of them are essential to its function in one or another aspect. The number these days is somewhere in the 60s of well-documented ones. At this time, it was more like 50. So that means that we have a big problem. We are now have biology as an information science. Information, why? Because of the hugeness of the genome. Information because we can't function without a computer, and with the combinatorial complexity and the relationships that we have to remember, it's hopeless. Even if you could sequence and assemble without a computer, it's hopeless to think about the biology without a computer today. The combinatorial complexity is huge. And finally, there is this problem that there's way more data there than we understand. We have, as biologists, what I like to call a NASA problem. NASA took all these pictures all over, and they're sitting as electrons in some computer somewhere, maybe Los Alamos for all I know, and no one ever looks at them, okay? Because we have no way to make any sense of them. People sample them. And we have that kind of problem. We have much more information than we understand. And part of that problem is that we're pretty good at computing, but we're not very good at communicating between the computer and ourselves. So to address this problem and more or less anticipating this problem, we started to think for the yeast genome, long before the genome was actually sequenced, we began to think about the question of could we organize the yeast data so that it could be computed upon, that we could search and find with reliability relevant data with a sensible query. And we started that when we started sequencing yeast, and we did lots of work using those data that were used to make the maps. I showed you the Mortimer map, and before that I showed you the coli maps. And those were, in fact, functioned as genome databases before there were genome databases. If you found a gene was mutant in your experiment, you went to look there to see if anyone had mapped that gene or anything near it and what was known about it, and the guide to the literature, it had all those elements. And so we decided to computerize that. And we decided, first of all, as did NCBI, that the place would be run by biologists, not people who knew how to run the computers, although it was important to run the computers. And we were very much taken with the fact that the point of this was to talk to the working biologist. This was not a compilation of data that could be taken down by command line by a bunch of experts and crunched upon, although that's an important function. This was for a simple query. And so I used to go around at the time that Genbank was moving, and there were other databases that should remain nameless, using the test. And the test was, I asked each database, what is hemoglobin? Or what do you know about hemoglobin? Or what do you know about HB2B? Okay, and if I didn't get an answer, I declared it useless. Okay, we of course didn't do that, and NCBI didn't do that. The whole thing, Jim's talk was masterful in showing you how much they were listening to what people needed and wanted. So what we ended up was making what is really a mini version of NCBI, but focused on something entirely different, which is what do the genes do? This is usage up to about, I don't know, 05 or something. Okay, so as soon as a lot of sequences became available, it became clear that we had a different kind of problem. And that different kind of problem was beautifully put by my friend Michael Ashburn. He said, biologists would rather share a toothbrush than a gene name. And this went double across species. Okay? So you ended up with situations like this. CDC 25 is son of 7 lesson flies. Talk about useless naming compared to let's say, E. coli, who look like heroes in this regard, right? This thing turns out to be RAS, right? And most of you, unless you're experts, had no idea that either of these things is orthodox RAS, right? So the idea was to produce a controlled vocabulary to describe the gene products and their functions in such a way that if a yeast biologist queried, blasted the world, and discovered that Drosophila had a gene, that what the thing they got back made some biological sense to them. Okay? That was the idea. Okay, so as soon as we started thinking about this seriously, this is the great thing about computerization. It sometimes makes you think clearly about the most basic things when you never had to before. In trying to get a computer to remember what a function is, it became immediately clear to us that we had to distinguish at least 3 aspects of function. One is the process, as defined here. The other is the actual molecular function, ATPase or something. And the last, of course, is the cellular component, or where. And to give you, again, to go back to the car analogy, if your Model T isn't running and you push it out of the way and you find a bolt on the ground that came from your car, and you look at the bolt and you ask Swissprot, what is it? And Swissprot says, fastener, 4-inch, actually 4 centimeters if it's Swissprot, okay? Okay? And so the question is, how much are you off better by knowing fastener 4-inch? You can see that, right? It'll give you the PI, it'll give you the molecular weight, and stuff like that. That's not so useful, okay? What you really want to know is what did it do for the car? Well, the molecular function may be fastener, but the cellular component could be the steering gear, okay? Or it could hold on the vanity mirror on the dashboard, okay? The biological process could be locomotion or primping, right? Okay? All right, and so you want this to be a structured vocabulary, and of course, Go, incomplete and imperfect as it is, has helped a lot in the bioinformatics, and it has helped hugely in the endeavor of individual biologists who want to know what's known. Okay, so I think I'm going to move on here, if my machine. Yeah, and then of course, we started to build tools that allow you to find the Go terms when you do some high throughput experiment or other, and that has helped a lot. I'm going to skip this. Okay. Now, so the intellectual impact of the genome has been, to a very large extent, one of unification. People used to laugh at this, including my friend who wrote the silly science article that Fink got excited about. It has also opened up a new frontier. And that new frontier is at this interaction level because the genome and the computer make it possible to do experiments at a different level than was possible before. And I'll give you an anecdote. When I was an undergraduate, I did undergraduate research. I worked in the lab of a great biologist, microbiologist, Boris Megasanik, and we were taking some organism whose name has been changed many times, and I don't know what it's called now. With all this proliferation of species, Jim, why don't they keep the old names the same? Anyway, this thing was called Aerobacter aerogenes. It's called something else now. Anyway, and the idea was that we soaked this organism as it was growing in tryptophan and asked what kind of new enzymes we could find. And the new enzyme we were looking for, of course, was something that did away with tryptophan, and we found something like that. And then Boris had me in his office and says, it's time to figure out whether this is specific, this induction by tryptophan of this tryptophanase or whatever it was. And I said, yeah, how do we do that? And he said, well, just measure a few other enzymes. So I measured ornithine transcarbamylase, and I measured some other NADPH using enzyme, and those weren't induced, and so we concluded it was specific. Of course, this experiment in the modern day is completely incompetent. It didn't make a lot of sense at the time, I have to say, but it was the best you could do. Now you can do much better. Before I show you any of this, I want to deliver another message, and that the message is about ourselves. In 1940 or so, a professor at Columbia, Tavis, had a bunch of people in a dark room, undergraduates at Columbia. And they were taking his course. And they were shown in the dark a number of spots of light for a second or less. And then they were asked to write down, A, how many did they see? And B, how sure were they? And what you see here is the confidence with which they saw how many spots of light. And as you see, this hypergeometric fall in confidence after the number 6. They were great at 6, and then it went away. Now, in fact, the rest of this paper is saying that even this is too confident. We're worse than it appears at this. The number of genes in the human genome is, I don't know, but it's in the tens of thousands, much bigger than 6. And that illustrates my problem. You get a result that involves thousands of genes. And how do you understand it when you have your brain, which is good at 6 and not bigger numbers? So you have to compute somehow first. You have to do something to show you the result. And this is now, I think, a major challenge. Not just remembering the data, but presenting it in a way that means something. So microarray is the example. And microarray data starts out as numbers, get reduced into a certain kind of picture, which isn't very useful. And then Mike Eisen figured out this picture, which is useful. And what you're looking at here is not a number, but a color. And the color is determined by how far away you are for the median for the ratio of expression between the red and the green. And this thing has been clustered. And so you can see that this set all have in common that they go from low to high. And those of you who are colorblind, you can see from blue to yellow, low to high. Okay, and you can actually remember what all the genes are, even if there are 10,000 of them, because you see this on a computer screen, and you can use hyperlinks. And if you have Go, and you have something which is called Go Slim, which just gives you a couple of words for each function, you can actually get some idea of the function. And if all of these genes all say ribosomal, all say translation, you say, gee, that's a biological result. Okay. We can do cell cycle experiment in which we synchronize cells, and what you see is that if you compare the synchronized to the unsynchronized, you can calculate some kind of Fourier score, and you can see these stripes. Okay? But suppose I didn't have the stripes. Suppose I didn't have the idea, the prejudice about that. Could I just look at internal data? And in this case, you can look you can cluster the data on the basis of some measure of similarity, and when you do that, you get a pattern of a pattern here. This is a dendrogram showing the Pearson correlation coefficients, and what you see here is that the stripes are visible. So you would have discovered the stripe without the prejudice. But even more interesting is if you look at the annotations for each of these sets here, these clusters, these deep clusters here, you see that they are functionally related according to Go. And if you read all the papers, you find that they're functionally related according to all the papers, because Go is bad, but not that bad. Okay? So now you can do a different kind of experiment. Different kind of experiment goes something like this. I have a hypothesis. Genes whose patterns of expression are strongly correlated are likely to function together. Okay? So the test is, I'm going to look at all these genes, and I'm going to ask if they are overrepresented in a particular Go category. Hypothesis 2, same thing for position. Okay? Both tests consist of just doing a simple statistical test. And when I do that test, I did this for a subset of this particular group for clarity. This is the Go graphical output. The different colors mean different degrees of enrichment. And here is 3 of these genes. The prior probability, the bootstrap probability that they would have gotten there by chance is between 1 in a million and 1 in 10,000. And for the other case, it's between 1 in 100 million and 1 in a million. And so we have a kind of inference that wasn't possible before. And it's very general, I should say. Here you can look at the patterns of gene expression with my collaborator, Pat Brown. We had a lab that did this kind of stuff for about a decade. And you can see all kinds of things. And I want you to notice 2 things. You can see that they're all different just by looking. The calculation has been done for you, but presented in a way, in a Tavis-proof way. You're not looking at any numbers, you're not doing any calculations in your head. You can just see it, because we are evolved to see things coming and going. You can do the same thing for tumors, and when you do it for tumors, you get a very good result right away. All the breast tumors, the purple ones, are all together, which means that they're more similar to each other than they are to other tumors, and so it's not very likely that you're going to have an ovarian tumor that looks like a breast tumor. You can do more than that. You can cluster now also the patterns of gene expression among the samples, and then you get arguably 4 subclasses of breast cancer. And you can do this again on somebody else's data set. So it's beginning to look very much like sequence information, this information. It's worth keeping, it's worth cataloging, it's worth annotating, it's worth having in Genbank or some NCBI thing. Which there is, and because I can now take somebody else's data if they're available, and I'll come back to that in a minute, and then see that the red ones are still together, the blue ones are still together, the light blue ones are still together, and the magenta ones would be together if there were more than 6 of them. This has a clinical consequence as well as a biological consequence, because in both data sets, the different patterns of breast cancer are characteristic of tumors with different lethalities. So now I can make a different kind of genomic hypothesis test. Now this is really a genetics experiment. The 4 cancer breast types, I argue, are different diseases. If so, then women who inherit one of the genes that predisposes to cancer should, at least by Occam's razor and by logic, might be expected to have the same tumor subtype. And so we can just do the same kind of thing that we did with GoTerm Finder. And what you see is that the BRCA1 carriers are all the dark red type. And the BRCA2 carriers, this is still an anecdote in this data set, because we don't have the genotypes of all of them, because BRCA2 wasn't. It's a long story, never mind. But you get the picture. Okay. So my last post-genome anecdote has to do with understanding, I got 5 minutes, okay, really what the classical, now it's called system biology. This is not a new idea, like all the others. Ira Herskowitz made this diagram summarizing decades of yeast work, of phage lambda work, and this is all still pretty much true. What's interesting about this diagram, besides its complexity, is the fact that all of these relationships are unquantitative. They're digital. Goes up, goes down. And you can do new kinds of data, like the mass spectrometry. It's now really possible to measure compounds. And if we can't understand metabolism at the system level, what will we be able to understand? You can do experiments in which you can follow not only all the genes, but all the metabolites. And when you do that, you get sensible results. If you use the same kind of bioinformatic approach on the amounts of a metabolite under, this is starving E. coli for carbon or nitrogen, this is starving yeast for carbon or nitrogen. Okay? And you get some really amazing results. Notice also that the dynamic range of this assay is 256-fold in each direction. So we're not talking about small differences here. This is not little noise. And you can't read these, it doesn't matter, because the result here, this is now a singular value decomposition or a singular component analysis. And what you see is that there are three major vectors, and the three major vectors are organism-dependent, metabolite-dependent. Most of you probably thought that was going to be the major class. Organism-independent, but metabolite-dependent. That's reasonable. But a whopping 42% of the variation in this experiment is independent of both metabolite and organism. What does that mean? It means that we all live on the same metabolic plan. That the connections, no matter what you want to say about what happened, the connections in these two free-living organisms that are millions of years apart, the connections between the metabolites are such on the building plan that when you starve for carbon, you get a stereotyped response that both remember. We can do experiments in which we find out things in yeast that remind us of things in tumors. It's not important what we found, but you can see that these curves are different, and so we interpret that as being something like the Warburg effect, which is that tumor cells waste glucose and normal cells never do in culture or in tissue slices. And so what we did was we looked for mutants, okay, that suppress this effect because we're still geneticists. We're doing basically what Rich wants us to do, is to investigate what's actually going on. And we do a lot of that, and we get mutants. Now, because it's the post-genome era and we have a chip on which we can find a single mutation in the whole genome, we don't have to go through a lot of rigmarole to find out where these mutations lie. And we found the first two mutations we found were in a gene of which we'd never heard of before called PPM1. And this PPM1 is part of a complex that has to do with protein phosphatase 2A, which is very highly conserved everywhere. And as you can see, it really does suppress the effect. This is the effect. This is the mutant. You can see we're back to normal. And so why is this here? This looks like ordinary microbiology, right? Not so, because we can now look at all these things that we got when we do a much bigger screen. And then when we ask for all the things that we believe in, we ask, where are they in the diagram of related genes that's derived bioinformatically from all the data that's around out of SGD by Olga Troyanskaya's group? What you see is that the gray ones are the ones we found, okay? We found PPM1 in here somewhere, and we found all these guys. And if you ask who is the center of this, it's CDC 55. And if you ask who are the closest relatives by all kinds of interaction methods to CDC 55, they include something that is the protein phosphatase 2A itself, something that is involved in mitosis, another thing involved in mitosis, something involved in entry into the cell cycle at the very beginning, something that's that is the other subunit of that, spindle assembly, Phosphofructokinase, remember this is all about wasting glucose. That is the central regulator of the glycolysis. Bud emergence, which is in the middle there somewhere, and glucose repression. So, by experiment, I blundered into a little knot of 50 out of the 6,000 yeast genes, 5,700 yeast genes, that are all stuck together. This is something worth spending a lot of effort to figure out. So that's the end of my story. I want to summarize only in the following way. I think the time has come to become much more serious about the things that are above Jim Ostel's line, above the proteins. What do the proteins do? What do the other elements in the genome do? I think only in that way are we going to get any real purchase on what is actually happening and real purchase on how complex diseases and gene interactions work. And to that end, we need to get much better at remembering this information and serving it up in a way so that someone who doesn't have a degree in biophysics can understand it, or at least look at it. And we've done extremely well. NCBI has done, I think, an exemplary job in getting us to there on the sequence level. But now it's time to spend comparable effort, at least, on higher up level. So this is basically to credit individual people. I think Mike Eisen needs to be remembered. I think this is better, yeah. Mike Eisen needs to be remembered because this visualization of gene expression, he was really the central actor. Kara Dolinsky is a major player in the Go business and the SGD business. And all of these people that I've had the pleasure to collaborate, especially Mike Cherry. Mike Cherry is sort of a, I don't know, soul brother of Jim Ostel. Same kind of thing. He got into computers when he was a graduate student who should have been doing something else. And I was so fortunate to recruit him. I knew we needed to do SGD. Would never have happened without Mike. And he now runs it, and it's a great thing. And Pat Brown, of course, invented the microarrays, and we had this fantastic collaboration. Ashburner and Judy Blake were co-conspirators in Go. I'm really a minor conspirator. Olga is sort of one of the new bioinformatics gurus. Josh Rabinowitz does the mass spectrometry at our place. And Mike Ashburner and Judy Blake, of course, and Annalisa is who I left out, was where the breast cancer stuff came from. And so I thank you again for the pleasure and the honor of haranguing you about what you should do next, and I'm happy to take any questions.
You said that part of the big challenge going forward is the ability to analyze high-dimensional data sets. I recently co-chaired a Keystone Biomarker Symposium where by the end of the meeting there was this sinking feeling that the community is creating a lot of red herrings by statistically overfitting high-dimensional data sets. And one of the examples that was used was one of the predictive breast cancer data sets from the Dutch group.
Right.
That data was overfit, and,
right.
And when you re-examine it, the effect is still there, but it's been exaggerated by 100% by the overfitting. So how do we avoid, going down that path?
Okay. First of all, you're right. In that Greek paper where he talks about the Dutch data set, the Dutch data set was the second data set we analyzed. Okay. Our effect, which is not done by overfitting but is done unsupervised, is completely there. Their effect, which is completely overfit, is not present in our data set or any other data set. That's the problem. Okay? I'm afraid that the problem is exactly the same problem that happens when you have a teenager and you give him a Ferrari. Okay? You have to know how to drive the damn thing at 120 miles an hour. The fact that it will go 120 miles an hour is interesting, but dangerous in the wrong hands. The problem, it really is that statistics, we teach undergraduates and medical students in particular the wrong math. They have no idea. This overfitting is not surprising. There's an endless statistical literature that says you can't do what they did. You can't mix the same data set and analyze it twice, and so forth and so on, right? Which they did, okay? So I'm afraid that what I have to say in this case is that they were less than fully competent. Now, the question you should be asking is, why did the New England Journal print the thing? That is much harder to explain, because presumably they sent it to someone who knows some statistics, okay? I told you that it took me 10 years to publish that negative result on manic depression, which now everybody believes. The reason was that my collaborators were optimists, and they kept hoping that by reanalyzing the data, by dredging through the data, they would find something that positive to publish, and they didn't have to publish bad news. Okay? Even though I was happy to have it published in the Proceedings of the National Academy. That's why they made me a member, I assume, is that I can publish things like that, okay? And I did, okay? The point, however, is that there is a natural human desire to have a good result, and the whole idea behind statistics is to be able to do that without bias. And if you look, and it's somehow hard to convince ordinary humans that multiple hypothesis testing requires a discount. That's where these guys all go wrong. So what can I do? I mean, my thing they wanted and sort of published as it was inherited, but it had a parent of origin effect. You can imagine where that came from, right? It was just a false positive, right? So I don't know what to do. At Princeton, we're trying to teach everybody statistics as freshmen in the hope that you know, we'll do our small bit to have one fewer optimist out there.
Any other questions?
That was a good question. Got more? Okay.
Well, thank you very much, David.
My pleasure.